Okay, Mendelian genetics. Gregor Mendel was a monk, an Austrian monk. And in the monastery, he was in charge of taking care of the garden. And with that, he started to be interested in inheritance and using the pea plant as a mode of studying inheritance and patterns of inheritance and those kinds of things. So genetics is the study of genes. Inheritance is how traits or characteristics are passed on from generation to generation. Genetic material, DNA, genes, chromosomes, is found inside the nucleus of a cell. Like we said, Gregor Mendel, the father of genetics, first biologist to use mathematics to explain his results using numbers, and that was different. All right, so Mendelian genetics introduction here. Genetics, branch of biology that deals with heredity and the variation within an organism. Remember, more variation is better, more healthy organisms and species. So you're looking at quite a bit of variety in the human species and a lot of different animals. So variety is a good thing. Chromosomes carry the hereditary information. Here's a chromosome on the right in the picture. And you can look at each little band as a particular gene or a trait that some individual carries. All right, and we know the central dogma from back in the day when with Watson and Crick and saying that DNA has the message that gets coded through RNA, and then at the ribosome, it's converted into a protein. So that's the sequence. All right, so Mendel's themes. Observable variation is essential for following genes. So you got to be able to see a change. Variation is inherited according to genetic laws and not solely by chance. So there are very specific laws to it. And then Mendel's laws apply to all sexually reproducing organisms. So it could be a dog, cats, elephants, lions, humans, whatever. So traits are passed on to the next generation. If you look at these individuals, you can see how they look very similar and they're from the same family. Chromosomes and genes occur in pairs. They're known as homologous pairs. You get one of the pair from your mother and the other individual in the pair is from your father. And they get passed on. Remember, those homologous chromosomes are what are being split in meiosis that we just learned about. So you only are having 23 in each of the sex cells, the sperm and the egg. New combinations of genes occur in sexual reproduction during prophase 1, where the homologous chromosomes can have some crossing over take place. And fertilization is when we have a sperm and egg combine. So Mendel and his pea plants. He looked at 7 different types of characteristics within the pea plant. He looked at the stem length, could be tall or short, dwarf. He looked at the pod shape. It's either inflated or constricted. A seed shape, smooth or wrinkled. The seed color, it was either yellow or green. The flower position, it was either at the end or out on the side. So lateral or terminal. He looked at the color, purple or white for the flower. And then the pod, he looked at whether they were green or yellow. It's a pretty simple plant and a pretty good plant to choose because it wasn't too complicated. He was a talented conductor of experiments. He used large sample pools, which is very important because, as you know, you can flip a coin 5 times in a row and get heads every time. But if you did that 1,000 times, a good chance that it would be pretty close to 50-50 heads or tails. So using a large sample pool is important. Use control groups. So he was able to double check his work and make sure the results that he got were accurate, and choose easy traits to cross. And so if you look at the picture here's what he wound up doing. He took the stamens from the purple flower, right? That's the male part. And he transferred the sperm-bearing pollen to the white flower. And then he allowed the seeds to develop. He planted those. He examined them in the spring. He noticed that there weren't any white flowers. Interesting. Genetics terms you need to know. A gene is a unit of heredity found on a chromosome, a little section of it. The genome is the entire set of genes in the entire organism. So if you think of all the different genes for humans, an allele is a form of a gene. So you can have allele, you can have the gene for eye color, but you can have an allele for brown or blue or green. It's a different form of that particular trait. A locus is a fixed location on a strand of DNA, a certain spot. Homozygous means having identical genes, one from each parent for a particular trait. So, for example, you inherited blue eyes from your mother and blue eyes from your father. Heterozygous means you have two different forms of that gene, so you wound up having brown eyes and a blue-eyed inheritance, one from each parent. Dominant is the allele that tends to hide or suppress the expression of the other allele. So brown tends to hide the expression of blue eyes, and recessive is the one that gets masked. Some other genetic terms. Genotype, genetic makeup of an organism. Again, does it inherit two blue eye genes or brown and a blue, or two browns? The phenotype is, what does it actually look like? That person has blue eyes. So monohybrid cross is just a cross between a genetic cross involving a single pair of genes. So we're just looking at eye color. The parental generation is the parents. So in this picture, you can see there's a white plant on the top. That's one parent, and a purple plant on the left. That's another parent. That's the P generation. The F1 is the first filial generation of offspring from a genetic cross. So all the ones inside the box, all the purple ones inside the box that are a capital P and a little p, those are your filial one generation. And then if you cross those purple ones on the inside and got another generation, that would be your F2 generation. So Mendel's experiments, parents differ by a single trait, crossing two pea plants that differ in stem size, one tall and one short. So you had an individual, if you look on the right here, one parent was a purebred for tall and the other one was purebred for short. Two capital T's are dominant traits and two lowercase t's are the recessive traits for the short. When that happened, what would you expect to see as in their first filial generation, the offspring of this parental cross? So once he did that, he noticed that all of the offspring that came up were of that hybrid variety or the heterozygous variety, the capital T, dominant, tall, and a lowercase t or the recessive short. That was all of the F1 generation. So inside the box, you would see 4 of those. And again, that's just simply an outcome, a probability of something happening. Okay, so then the next thing he did was he took those offspring. This is the same cross right here, showing you how to use the Punnett square. Again, you put the parents on the side and the parents on the top, and then you add the individuals inside the box, and you can see what happens. All right, so determine the genotypes of the parents, write down your cross, draw a Punnett square, and then add into the boxes. Okay, pretty straightforward. Split the letters of the genotype for each parent and put them outside the P square. Determine the possible genotypes of offspring, filling in the Punnett square, and summarize the results. If you let the F1 generation self-fertilize, the next generation would be what you're looking at here, a capital T with lowercase and a capital T with lowercase. And now if you look inside, we get the short variety back again. Remember the F1 generation, they were all tall. Now, because they're recessive, two small t's inherited together produce a short plant. So the F2 generation is what you're looking at inside of here. The key to the Punnett square, determine the gametes of each parent. So this one right here, each one will go in, because of meiosis, remember they split the chromosomes. So we're gonna split the genes. One of these goes into say an egg, and we'll put it here, or a sperm. The other one's gonna go into that sperm. Split these, form an egg. One goes into that, one goes into this one, Now, when you have fertilization, the sperm and this egg will produce that individual. This sperm and this egg would produce that individual. Obviously, they're all the same. Phenotype, what it looks like. Okay, again, what could be the shape, the color, how tall it is. Does it have hair, not hair? Those are all things that you can actually witness and see. So that's your phenotypes. Genotypes, or what are they actually made of? So a purple can be a homozygous purple, right, where you would have two capital. It could be one of those heterozygous purples where you have one of each. And the capital P is the dominant trait. It's purple and it hides the white recessive trait. Or you can have a white flower, that's its phenotype, with a genotype of two lower P's. So here's some more examples. This one's with flower color, purple being dominant, white being recessive. If you take the purple homozygous and you cross it with the purple, the white homozygous recessive, right here you see that, purple homozygous dominant, white homozygous recessive. The only offsprings that they potentially can have is a purple hybrid plant. And a couple rules here. The dominant trait's always written first. So a capital is always written first, and then the lowercase, the recessive, is always written second. So if we cross these together, a purple hybrid with a purple hybrid, again, we wind up getting that 3-to-1 ratio, 3 purple to 1 white, or a genotypic ratio of 1 pure dominant, 2 hybrids, and 1 pure recessive. So principle of dominance, one allele masks another. One allele was dominant over the other in the F1 generation. Principle of segregation, when gametes are formed, the pairs of heredity factors or genes become separated so that each gene, sex cell, egg and sperm, receive only one kind of gene. That happens during meiosis. And that is it.